Okay, Thomas, let's have a bit of a chat about brain connectivity mapping. Do you want to set the scene for what this actually means in the context of whole brain light sheet imaging compared to other modalities such as fMRI, for example?
Yeah. I think connectivity is like the topic where um, the perfect umbrella term, so to say, because people can think of connectivity in many different ways, but at different scales, so to say. So sometimes to the level of the synapse, sometimes to the level of axonal pathways. I think within the light sheet space, so what we can do using a light sheet microscopy is more at what is considered the mesoscale, so to say. So not quite a single sort of a synaptic bouton resolution, but not quite, exactly what you mentioned, like a MRI scale. So it's like you distinguish cells, but you can also see individual like axons in the brain and follow them and see both where they start and when they end. So I think with Lightsheet, what you can aim at expect seeing using a light sheet microscopy is both the cells that you're staining, and usually the staining is achieved using usually is viral transfection. So you would, as I mentioned before, like the inject stereotaxically like an animal in the region of interest. Usually it's an animal that's a transgenic animal. So you specifically, you would infect a subset of cells in an area that you're injecting, and then basically label these cells with a fluorophore. So the cells would be fluorescent, and you just simply can trace them in 3D using light sheet microscopy, because you would clear the brain, stain for the specific fluorescent protein of interest, and then follow them across in volume, right? And what you should expect is to see both the cell itself, so like the body of the cell, that of course we're able to resolve, but also the axon and the dendrites, meaning that you can actually follow the path that the axon is taking. And this is useful, but of course has limitations, because then what you're able to quantify is, or where does this cell exactly project to? But that combines both the information of sort of like the axon itself, it's the pathway that it takes, but also more importantly, where it terminates, right? Because ultimately, this is what most people are interested in, is where do a specific cell type project, but also where do they release the specific neurotransmitter or neuromodulator onto its target, right? And so these are two separate things in a way, because you can, with light sheet, you can both trace the axon itself and the terminus in a slightly different way, but we can discuss that in a bit. Yeah.
No, absolutely. And then I guess what's the differentiator between the tracing projection and then doing like full connectivity mapping? Obviously, there's the fundamental difference that you're looking at something in particular rather than like from a much wider scope. But technically, how does that look?
Yeah, exactly. So if you think of how people study connectivity within more like the MRI space, that would be more like diffusion MRI and things like this. So what you would be looking at is sort of the main highways in the brain, right? Like which brain areas connect between each other, the directionality of these connections, like what is exactly the pathway that the axons take across the brain to reach their target. But ultimately, the key missing information in these data sets, outside of course, they have a limit in their resolution capabilities. But the main sort of pain point of these data sets is that you simply don't know, you don't have cellular specificity, right? So the only thing you can really claim is this brain region connects with this brain region, right? Either physically or functionally, if you think of like functional connectivity data also. But you don't know, like you cannot basically put a name on like what is the actual cell type that is sending that projection, right? And this is where sort of you have that trade-off between MRI can give you the big picture at a lower resolution, and then you can also go very precise, like more like cryo-EM reconstruction. So do very, very thin sections of brain tissue and then reconstruct to the single synapse level connections between a neuron and its target. Light sheet sits a little bit in between. Again, it's like the sweet spot between we have the full picture, we cannot fully resolve to the level of individual synapses, but at least you across the entire brain and sometimes the whole CNS even, you can actually follow the main pathways of axonal projections from the source to the target, but in a cell-specific manner, right? So we don't have to restrict ourselves to just, these are the highways, but you can actually make more precise claims as to this specific cell type that is defined by the genetic expression of this marker sends projections here and not there, right? So it's very useful for doing something called basically dissecting neural circuits, where you would do, you can do multiple injections of viruses in the brain that maybe infect different cell types in the vicinity in the same brain region, but you will see that these cell types actually project in very, very different, not only target areas but also patterns in space, right? And these are informations that are completely masked by things like diffusion MRI studies, right?
And how does Vibraint run a connectivity study? Like, talk me through the end-to-end workflow here, and I guess also perhaps something we've touched on briefly before about some of the parameters that you guys will refine or tune to.
Yeah, so I think there's the typical study would be, again, that people run their animal work in-house, right? So they have, depending on what the question is, right? If it's more general, then perhaps they use just wild-type mice, but usually it's a transgenic animals model, right? So we don't provide that, but it's people have these animals in-house. And the vast majority of experiments would either involve some sort of tracer, that's usually like things like fluorogold or cholera toxin that are fluorescently labeled or that you can visualize directly after clearing. That is one typical way of visualizing them, but to be honest, most of cases use some sort of virus. And usually these are either adeno-associated viruses, so called AAVs. These are either called the constitutive, so meaning that they just express in any cell that they infect. Of course, they have, depending on what capsid you're using, maybe they're preferentially infecting neurons or glia or excitatory neurons versus inhibitory neurons. But generally speaking, they just, whatever they infect, they will express the construct in these cells. But you can also have called the conditional viruses. So if you have a transgenic animal and you inject that conditional virus, then the virus will only express in cells that express a specific gene, for instance, right? So let's say a cell type expresses gene X, then the virus infects that cell and it will express itself. If the cell doesn't express the gene, then hopefully the virus does not express itself, right? And so people would inject either, again, tracers or viruses in the tissue, let the virus express itself. Usually it takes a couple of weeks for viruses, right, to properly express themselves. And then extract the tissue, send it to us, and we would actually stain for the tag that whatever the virus was expressing, right? So generally speaking, it's a fluorescent protein. So there's two strategies here. Either we preserve the fluorescence itself. We usually, I would tend not to do that because like a lot of the techniques we're using is actually killing the fluorescence of native proteins. So we would stain against them. So you come with an antibody against, for instance, GFP, the green fluorescent protein. And this has two advantages. First one is you actually amplify the signal, right? So let's say the expression, it's usually not so much the case for viral tracing, but you in any case have a much cleaner signal. Basically, if you stain against GFP, you can then clear the tissue, and you basically, the fluorescence you're visualizing is you indirectly labeling the actual GFP protein, right? And you visualize this in 3D. Usually, you then see, of course, the cell itself, the cell that was infected by the virus, together with its projections across the entire brain. But the really cool thing is that nowadays there's a little bit more sophisticated constructs that combine in the same virus. You can both label the projection itself as well as the synapses. So, and this is very powerful. You can do this in 2 separate colors. So for instance, one example that I can give you is, for instance, using with GFP, you can see the projection itself. And with RFP, like the red fluorescent protein, you would see, for instance, it's fused to synaptophysin, for instance, which you would actually visualize the synapse itself. So within the same scan, the same tissue, you both have the information about where is the cell that was infected, where does it project, and also where is actually the content, either neuropeptidergic or the actual neurotransmitter being released in the target site, right? And so these we can do simultaneously in 3D. You just stain for the two and basically visualize this in 3 dimensions, right? So you can trace both where things go and where are things released. And these are complementary informations, right? Because one gives you the source and where it goes, and the other one, the very precise location of where the target is. Yeah.
That was a really good description of how that's run. And then so that there covers the part that I guess a researcher would do themselves. Where does your services bolt onto that and the analysis that you do on top of that?
Yeah, so as I said, they would run all of this in-house. They collect the tissue and then they send it to us. We would then stain for the specific, like whatever tag that virus had, right? Do an immunofluorescence against it, clear the brains, scan them, of course, register them to the atlas, and then run segmentation. Here, the segmentation is, of course, more involved because depending on the type of signal you would be segmenting, you will most likely want to both segment the axons themselves, right? So it's sort of like tracing filaments across the tissue, but it's really important to realize that this is close to like the resolution limit of what Lightsheet can do. So I don't think it's fair to say, to claim that we can segment individual axons. What we're aiming for here, again, is some sort of intermediate resolution where we can look at the general projectome of these cells, right? We're not aiming really at saying this cell projects here, but more like this collection of cells projects in those areas of the brain. The cool thing, again, is that you have the whole brain readout in an unbiased way. So, right, so you can see the general projectome of a specific cell type across the entire brain. But more importantly, you can, again, pool animals, average them together and look at, how stable some of the patterns are across animals, right? What is consistent, what is not consistent, and then, of course, draw conclusions from this. So one aspect is segmenting axons, and the other one is segmenting maybe the fused synaptophysin, for instance, fluorescent proteins. This would be puncta, right? So similar a little bit to like how we do CFOS mapping, where we're basically quantifying nuclei in space. But in this case, you would be segmenting puncta, which are basically the synaptic connections, right, of these neurons onto its target, except that the puncta are much smaller, obviously. So this has its own challenges. Of course, resolution is also quite important here. But generally speaking, the way we image these brains is a slightly higher resolution than when we usually scan other things, right? Because the type of things we want to resolve spatially are more the details are much higher. We need higher resolution imaging to actually get this level of detail. But down the line, the output that you get is, of course, depending on the type of virus or tracer you're using, right, would be a whole brain map of where the specific cells that you infected project across the entire brain. And usually that involves some sort of averaging. And then statistics are, of course, important, but also sometimes usually optional in a way where the control group would be just the same virus being injected in an animal that hopefully has no infection, right? Where the virus should not infect any cell because typically the transgenic construct of that animal does not allow the virus to express. So if there is expression, it's obviously, unwanted expression. But it's so usually the statistics are more to remove basically background staining that you would see in animals that should not have any staining to be seen, if that makes sense. Yeah.
Yeah, that absolutely makes sense. And I've got a couple of questions off the back of that. The first one's a bit of a personal side question. When you're talking about imaging in high resolution, is that the actual raw imaging itself is in the high resolution, or you are compressing the output less?
Yeah, so the issue is in terms of segmentation, right? If we discuss segmentation, then if you think of you want to segment an object in an image, right? You want to be sure that you are actually able to even segment that object in your image, meaning that you have enough granularity of information to be able to separate two similar objects in the same image. If you think of like CFOS, right? You have two spheres in space. If you image at a resolution that's quite low, then the two spheres will tend to merge, right? They will make like sort of like this beaded necklace shape. And then, of course, you can maybe after the fact, right? Separate them in the segmentation process, but ideally you want to avoid that, right? So you want to select this sort of sweet spot where you image at a resolution that allows you to separate the objects that you're trying to segment as much as physically possible. That said, axons are typically maybe a couple microns in width. So this is really the limit of what light sheet can do. So when I was saying higher resolution, basically maxing out the resolution of what light sheet can scan at. Also relative to the actual physics of the light sheet itself. At some point, you're limited by how thin that sheet of light that traverses the sample can be. This is usually in the order of magnitude of a couple micron, like 1-2 micron. So this is really like at the limit of what light sheet can do, and that's why I would be careful of saying that we segment individual axons, but more we just segment broad axonal projection patterns, right? So there are two separate sort of questions. Some people are very interested in, I just want to know the general pattern, right? I'm more interested in what are the brain regions that this specific cell type projects to. Whereas other people maybe are more interested in, I want to know where the specific neuron projects to, right? And these are completely different problems. The latter is very difficult to do with light sheet with classical techniques. So some people are able to do this, but this is more on the experimental side where things have to be done in a certain way. So you would maybe do a very sparse viral labeling experiment. So you would inject a virus in a way, tweaking the concentration and so on, so that stochastically you would label maybe only one neuron, right? Then, of course, you can reconstruct the whole projection of that neuron. But more often than not, when people talk about viral tracing or just tracing experiments in general, you're infecting dozens, if not hundreds of cells simultaneously in the same area, right? So we're really looking at population level segmentation of the information here.
And this might be a stupid question, but if you, and you said you didn't want to overclaim on being able to distinguish like an individual axon path, how do you go from, like, if you say you can't determine that exact axon path, then how does that work with still being able to sort of generalize it across like a wider brain atlas if you can't necessarily like trace it exactly? Can you explain just how that works?
Yeah, I think, of course, this has many assumptions, right? So again, this goes back to what is a cell type and so on. But if you think about it, let's say a concrete example, right? You want to trace neurons that you're interested in a specific subpopulation of cells in the, let's say, amygdala, central amygdala, that expresses a gene called PRKCD, right? So you take a mouse line that's PRKCD3, for instance. You have a genetic lever that allows you to come in with a virus, infect these cells specifically. right? And now you make the following assumption. You're like, if I infect the cells expressing that specific gene, these cells are coherent enough, right, within the amygdala, so that, again, I assume that they all project in similar brain regions across the entire brain, which might be true or might be completely false. But you're aggregating that information to, the only claim you can make is that this is the general projection pattern of cells that express the gene marker in that brain region, right? It's not it's not excluding the fact that there might be subpopulations within that coherent set of cells. Let's say maybe there is a subpopulation of these neurons in the amygdala that projects primarily to region A, and another one that projects to region B. That, of course, you cannot do unless you have a better genetic, marker that would define that population better. But the idea here is, of course, this is limiting in a way, but the idea is that you want to just look at the general pattern of projection of this restricted cell population in that area. And the important thing is that you look across multiple animals then, because you want to aggregate that information into one sort of average map of how this looks, right, across the whole brain. And generally speaking, it's quite good, I have to say. It's usually quite reproducible across animals. The tricky thing is that these type of experiments usually involve a little bit more animals than, let's say, pharmacology and doing whole brain CFOS mapping and things like this, simply for the fact that, there's variability in how precisely people can target certain areas, the rate of infection, like, how broad the injection is. Sometimes maybe they infect thousands of cells, sometimes hundreds. So of course the pattern might look very different, but it's maybe not different in terms of where the projections go, but more in terms of like how dense the projections are, right? Because you're just infecting 10 times the amount of cells. So these are also very important things to take into account, like how reproducible is your setup, right? How cleanly can you inject in the same area, infect reliably? Again, it's impossible to have exactly the same number of cells, but at least, like having something that is comparable across a group of animals that you're trying to compare against.
Yeah, thank you very much. That was a helpful explanation. So Vibraint offers graph-based functional connectivity analysis, right?
Mm-hmm.
What does that layer add on top of raw region-to-region signals?
Yeah, I think the really powerful thing, if you think of connectivity data, again, for me, connectivity becomes useful once you have a lot of that information, right? You just need to gather enough information to build like a general understanding of how cell types just connect across the entire brain tissue. This becomes extremely powerful in considerations like what we discussed in previous episodes, like pharmacology, right? If you have recruitment of different brain areas, they might be part of the same circuit or not, right? But this is something typically that you simply cannot know without knowing the connectivity, the underlying connectivity, right? So let's say that you first run an experiment where you see that pharmacologically region A, B, C, and D are recruited. In fact, using some of the tools that we developed, maybe you can go a step further where you say, well, it's very likely that the cells in region B are this specific subpopulation of neurons in the brain. Well, then if you have access, right, if that data set exists, or you can generate that data set yourself, if we know the connectivity of this specific underlying population of cells, and the connectivity actually matches spatially the pattern of activity that you see from the previous experiments, then this becomes quite powerful, right? Because you can start to make non-trivial, inferences between the two completely different modalities, right? If the connectivity can recapitulate activity, of course, this is correlative evidence still, but it becomes more and more solid moving forward for you to make better decisions and interpreting the data further.
And in terms of turning this into a 3D connectivity map, do you have an example of a projection or circuit that would be invisible in 2D, but only really like comes to life once it's in 3D, just to put it into perspective?
Yeah, I think many. Many, if not maybe most, because some specific cell types that I can think of, some connectivity is local, right? So maybe you can argue in that case 2D is good enough, right? So if you think of maybe interneurons in some parts of the cortex or some specific cells in the hindbrain, the connectivity is local, meaning that there's no long-range projecting connection between that cell type and cells in the brain that are located far away. It's local connections. 2D section probably captures this really well. A beautiful example, perhaps, is neurons in the cortex that are known to control locomotion. If you think of this, these are cells that are located deep inside the cortex, so on the surface of your brain. But some of them project all the way down to the lumbar spinal cord, right? So the axon goes down all the way from the brain to the very tip of your spinal cord. You simply cannot capture this on a 2D section, right? And this is maybe the extreme example of that. But more often than not, projections are, complex 3-dimensionally. You cannot capture this from a 2D section. Of course, nowadays also the alternative technique people can use to study this is serial sectioning. So you would actually take like the brain and simply just cut it, do serial sections of the brain, but scan as you go. So you scan the surface of the brain and reconstruct in 3D as you go. That is, of course, quite powerful too. It's just that you usually quite cumbersome, right? You have to cut, then the image reconstruct in 3D. You basically, as you cut, you build the 3D volume. But essentially, it's the same, right? As same if not similar to what Lightsheet is doing. It's just that we actually don't need to cut at all, right? You just stain for whatever you're interested in, clear, and then visualize in 3 dimensions. Yeah.
Nice. And again, referencing previous discussions, we've mentioned, we've spoken about CNS Voyager, and you indicated that connectivity is something that is in the roadmap to come to CNS Voyager. So do you want to speak to where connectivity sits in regard to CNS Voyager and what your vision is for that?
Yeah. So right now, again, in terms of connectivity, we would rely solely on publicly available data. It's not like there isn't any, but it's limited, right? So I think there's 2 ways to move forward with this. Is either, if new data sets come out, we would happily integrate them in the platform to extend the capabilities of what connectivity can bring to the table as to interpreting these complex data sets. And the other way around is maybe we can actually invest in that effort and also either collaborate or generate some of these data sets in-house for maybe at the beginning more targeted questions, like, a very specific node in the brain we would like to explore in terms of connectivity. But basically slowly building these data sets in the viewer as we go, either ourselves in collaboration with someone or basically just making use of what's available currently out there. But I have to say exactly currently it's in the mouse brain. There are many data sets that exist, but they usually come in 2 flavors, right? One is exactly this more mesoscale type of imaging where it's 2D reconstructed sections using viral tracing, which is quite powerful. There's a few scattered Lightsheet data sets that are available. But also a lot of data sets are single neuron reconstructions, right? So one brain would be one neuron, and it's amazing, but it's also not really scalable, at least at our scale, right? So that would for sure not be us generating these data sets, but more, whoever comes up with these amazing data sets, we would be more than happy to integrate in the viewer and make use of them.
Fantastic. Thank you very much. Really interested to learn about it and excited to see what that's going to look like once it's in CNS Voyager as well.
Yeah. Thanks.